We're here to interview David Hibbett. And David is a long-time student of mycology as far as we're concerned. And one of my big regrets was that I thought maybe David would come to Baton Rouge on a postdoc, but instead he stayed at Harvard, which was a really good decision for him. And so we want to ask David some things like, where was he born? What was his family life like? How did he get influenced to go into mycology, and where he went to school and all? So Dave, where were you born?
Okay, I was born in Cambridge, Massachusetts, and grew up in Arlington. My father was a faculty member at Harvard, so he was a colleague of Don's, which turned out to be a very important factor in my life later. I went to public school in Arlington. I also spent a couple of childhood years in Japan. I went to first grade in Japan. Then I went to UMass Amherst, where I was not a particularly good student. It took me 5 years to finish up, including 2 summers at the U of Michigan field station. Then I took a year off and went to Duke, where I was Rita Svigali's first PhD student.
Okay, so you took a year off after your time at Massachusetts.
Yeah, I took a year off and I worked as a technician for the Army Corps of Engineers doing water quality monitoring, counting phytoplankton samples in polluted rivers all over New England.
Huh.
You know, students always ask me, I don't want to go straight to graduate school. Is that bad? Do you think it was good for you to work for a year?
Oh, it was fantastic.
Yeah.
It was one of the greatest years of my life because I was out of school, so I didn't have those obligations, and I felt like I had a lot of money. I didn't, but I felt like I did. And then after I was accepted to Duke, I felt like I had everything all set. My future was certain. I was very relaxed. I was completely unrealistic about what graduate school was going to be like or what my career trajectory was going to be like, but it was a magnificent year.
Good.
Yeah. You mentioned your father, and I will just insert the little piece that as a young assistant professor at Harvard, I was assigned by somebody, department or otherwise, to the FAS, Faculty of Arts and Science Library Committee. And I showed up at the meeting and we were introducing ourselves, and I introduced myself as a mycologist. And this man next to me turned and said, you know, my son is a graduate student studying fungi. And that was your father, of course. Yeah, so that was my introduction, both to you, you know, I didn't know you at that point, and to your father.
Well, it makes sense that you would have been on a committee together because you and he are bibliophiles. As am I suppose.
You are now.
Yeah.
Yeah, you are.
Yeah. So you went to Duke.
I went to Duke, not to study fungi, actually. I was a botany major at UMass. I took all of the sort of traditional ologies, botanical ologies I possibly could, and as I said, I spent 2 years at the U of Michigan field station.
Had you had the Bigelow's courses, Bard and
Yeah,
Bigelow?
so I had Howard Bigelow taught a sort of a 1-credit Mushrooms of New England course, which I took, and it was fun, and I thought mushrooms were cool, and I liked Howard and Margaret Bigelow, but I had by no means decided to be a mycologist. I also took Bob Fogel's Ecology of Forest Fungi course in Michigan, which was really ecology of ectomycorrhizal fungi. And I liked that also, but what I really liked was courses on bryology from Howard Crum, who was one of the distinguished American bryologists. So I chose Duke because Duke had a very broad program in botany, including bryology. Lewis Anderson and Brent Mischler were there at the time, and they also had phycology, which I liked, and obviously plant things. So I headed off to Duke intending to study plant systematics. And Ritas and I actually probably arrived in Durham within a couple of weeks of each other because he was just starting that year. And I knew I wanted to do molecular systematics, and Ritas had the only molecular evolution lab in the botany department at the time, that was 1986. And Ritas was incredibly generous with his laboratory resources and his knowledge, and he opened the door and he let me come in and try to work on plants. There was my office mate who was also working on plants was in Ritas's lab. There was a woman working on lemurs, Ann Yoder, very successful
Do you know,
zoologist.
I knew her at Hope College. She was a student at Hope College when I taught there.
She's a star. And,
Yeah.
and also the person who would become my wife, who was studying red algae, my wife Margaret Carroll, we were all in Ritas's lab working on things other than fungi. I was trying to study pitcher plants, and because I thought they were cool, which they are, and I drove all over the coastal plain trying to collect pitcher plants and filled our freezers with them. And those were in the days before PCR, so I was trying to get high quality DNA and RNA out of the pitchers, which proved impossible. And about a year into this, I just gave up on the Sarracenia project. I was very depressed. Everything was just, going to pieces. But I'd always been into fungi, so I thought, well, I need a new project. I'm in this mycology lab. Mushrooms are cool. I'll just switch to fungi, and that's what I did. I just stumbled into mycology by virtue of failing at being a botanist. And then a little soon thereafter, we got PCR in the lab. And if PCR had been available when I started grad school, the Sarracenia project would have worked. And I might be a plant systematist now, but I consider myself immensely fortunate to be a mycologist, because I feel like well, first of all, it's just a really fun field to be in with wonderful people and great biology and great problems. But it's also a small enough discipline that, I feel like I was actually able to do something useful, whereas, plant biology is a much bigger field.
Well,
so it worked out.
Small in number of people who study fungi, but big in numbers of fungi that aren't studied.
Yeah, right. Huge in scope and in importance, but underpopulated in terms of researchers.
So what did you do for your PhD when you decided?
Right. So I picked a genus Lentinus, which turned out to be a good, lucky choice. I was reading some of the works of some of the great mycologists, so Rolf Singer, David Pegler, Corner, and they all seem to be incapable of agreeing on the generic limits of Lentinus or Lentinus and Panus. And then Scott Redhead and Jim Gins had looked at the decay chemistry of these things, and they split out new genera. It seemed like a classic taxonomic conundrum. So I decided that I would try to work on Lentinus, both just to do sort of straight ahead systematics, understand the generic limits, but also there were these anatomical hints that suggested that Lentinus, which has gills, of course, was related to polypores. So at the time, gills versus pores was a huge big deal in fungal taxonomy. And so the idea that you could have closely related things with gills and pores really cried out for some work. The Shiitake genus Lentinula had also been pulled out of Lentinus, so that made it interesting. And it worked out really nicely. It turned out that what Pegler had called Lentinus was split across not just multiple genera, but multiple orders. So it was an early demonstration of the power of molecular techniques to resolve relationships in what we now call agaricomycetes. And again, it was good luck. I mean, well, it was good luck, but also I had to do a lot of reading to find the problem. Sure. But it was a good choice.
And then you finished up there, and did you go to Japan then?
Yeah. So so my project, the amount of molecular data that I collected in my PhD, you wouldn't even get a single poster at an MSA meeting with that amount of data now. It was such a small contribution, but it took me nonetheless. It took me 3.5 years.
Well, it was a good start, and it answered a question.
Right, but my point is that I did my PhD in 5 years. A year of that was spent on this failed Sarracenia project. So 3.5, 4 years of work on molecular systematics of Lentinus. It was really hard to get molecular data back then. We were running acrylamide gels to do DNA sequencing, and reading the autoradiograms by hand and all that stuff. So we had it tough. And so I'd put all of this energy into molecular systematics, but by the end of my PhD, I felt like I really didn't know that much about fungi. I didn't really know that much about morphology. I've always wanted to be the kind of mycologist that can go out in the woods and know lots of mushrooms, and I'm still not, and I certainly wasn't then. So I decided I wanted to do a project that was focused on morphology. And in particular, I wanted to focus on development, because I thought, morphological evolution is developmental evolution. So I did a morphological evo-devo project in Japan at the Tottori Mycological Institute. I had gotten a grant as a graduate student to study Japanese. The NSF for a while had a program to encourage American scientists and engineers to study Japanese. Incredibly visionary program. So I followed up that with a grant from the Japan Society for Promotion of Science to go for a foreign postdoc to Tottori, where I worked with Aki Tsuneda, who is a great electron microscopist. And Tottori is supported by the shiitake industry, so they really know how to grow mushrooms in culture. And I wanted to grow the things that had been, lentinus and things formerly classified in lentinus in culture, to look at ontogeny of the gills, because the molecular work had suggested that those gills were products of convergent evolution. I wanted to look at development to see if that corroborated it, and to see if I could learn something about the pore-gill switch. So we also looked at the development of the poroid relatives of lentinus. So I completely switched from molecular systematics to developmental morphology. And I'm really glad I did, because I thought the work that came out of it was, at least to me, it was informative. I learned a lot about morphology and fungal development. But molecular systematics was just going through the roof at that point. So strictly from a career perspective, it might have been smarter for me to have carried on with molecular systematics, because I kind of got in on the ground floor on that. But instead I took this giant veering detour, both in terms of the discipline and geographically. I moved to Japan for a year. And it was a wonderful experience. Margaret and I went and lived in Japan, and it was just, so fantastic to be there. And in this, my mother is Japanese American, so that was also important for me.
You didn't have any relatives then?
Oh, I do.
Oh, you did?
Yeah, I did, I did and still do in Japan and in Okinawa. But it was a hard shift. I was picking up a new discipline, kind of a weird area. Fungal development is, it's a little esoteric. And I moved to Japan for a year, and I had a very hard time finding a job, because it was pre-internet. Every now and then people would fax us. They would fax us copies of job ads from the back of Pages of Science, and I would apply and get no response. I actually did get one interview while I was in Japan, but the job didn't pan out. So Margaret, my wife, got a job in Massachusetts at Framingham State College, now Framingham State University. So she preceded me back to Massachusetts, where she had a job. And since I had no job to hurry back to, I decided to go to New Guinea on my way back to North America. And I went and I had a great time and collected some material that ended up actually being useful in research. And then I landed in a heap back in Massachusetts. I followed my wife back to Massachusetts, where I had no job and she had a job, and the job search had been a disaster to that point. I was very depressed, and I'm very glad you're involved in this interview, because Don played a really critical role in my career at that moment, because you offered me a position as associate of the Harvard Herbaria, and you gave me an office in the Farlow, and some excellent letterhead from which to write letters of application. And I sat in the Farlow for part of that winter of '92, '93, and wrote papers and tried to regroup and get my act together. I taught a microbiology course for nurses at Framingham State. And then at that time, Michael Donahue was coming to Harvard, and I remember writing to him and saying, dear Dr. Donahue, and I'd met him at a Willie Hennig Society meeting, so I had met him, but I wrote to him and basically just asked him to hire me, and miraculously he did, and that started, and then I had this fantastic postdoc, but you played a key role, and I am forever grateful to you for making that possible.
Well, I'm glad it all worked out. But then you worked for Michael and you had to set up that whole lab, right?
Yeah, well, I think Michael hired me because I had experience in molecular systematics, and so, and I got the feeling that Harvard had moved heaven and earth to bring Michael, and resources were not a problem. The lab was incredible, and I spent 6.5 years in HUH as a postdoc in Michael's lab. And Michael Donahue, of course, is an angiosperm systematist. I've always felt a great affinity for plants, and so working in the Donahue lab was wonderful.
Well, and Michael had an affinity for fungi too. He liked to think about fungi.
He did. He sort of became a patron of mycology. His brother is a shiitake grower. And Michael ended up actually giving us the Carling lecture when we met at Asilomar one year.
That's right.
So it all worked out very nicely. But then,
And his brother actually he worked for Mycology.
oh yeah, I didn't know that.
Yeah.
Anyway, so it took me a
think
long
you did.
time, but I finally got a faculty position at Clark University in Worcester. Along the way, my wife and I came to the conclusion that we were going to stay in Massachusetts for the long haul, and so I stopped looking at opportunities outside of southern New England, which, again, from a strictly careerist perspective, that limited my options. But my family is from Massachusetts. Margaret's family is from Massachusetts. She has a very good job at Framingham State. My children got to grow up knowing their grandparents, so it all worked out, but certainly not a straight line, and a lot of anxiety along the way.
So you became department chairman at some point.
Just interim for 1 year. Yeah.
Okay. I thought it was longer than that.
Nope, just 1 year.
Mm-hmm. And then, then you went. You got a Radcliffe Fellowship.
Yeah. The year before last, I was a fellow at the Radcliffe Institute, which was absolutely wonderful. The year went by very fast, but it was great to be able to spend time interacting with, well, there were some scientists there, including some evolutionary biologists. Axel Meyer and Jihang Yang were there, so that was, and I knew those guys in advance. But, mainly people from the humanities, social sciences, independent artists and journalists, so it was a great year. I worked on my current major project, which is a book on fungal systematics for a general audience or an educated lay audience, which is still nowhere near complete.
But I think it'd be good for students too.
I hope so.
Yeah.
Yeah.
A lot of students.
Well, Meredith was also a Radcliffe Fellow.
Yep.
We had this all planned with Meredith, that she was going to come and we were going to do things and everything was going to be wonderful. And then I was asked to be interim dean of Harvard College. So Meredith spent the year with me as interim dean, and we
Often missing from the table at lunch.
Missing, yeah. But
Anyway.
we had a good time.
But it was.
I mean, we both all did.
Yeah.
In
No,
your
it's a
time
very
there,
good
it was
experience.
great.
Yeah.
Yeah.
So, anything else you want to tell anybody that will watch this video?
I don't know. I mean, I don't feel like we've really talked about science all that much.
No, and you know, that would be a good thing to do, because some of the things that you've done recently are really interesting.
Thank you. I think my work has been integrative, and I've always done what I've always been very spoiled. I've done exactly what I want to do. So I wanted to do molecular systematics. I wanted to do development. And I've been able to focus on the things that interest me. I've done a little paleomycology, and I think molecular phylogenetics is probably what I'm that's the majority of my work. It's probably what I'm best known for. But I've also, I've really benefited from the advent of genomics, and I've had wonderful collaborators. One of the great things about the genomic revolution is that it's brought people together that previously wouldn't have necessarily collaborated, because the data are so accessible now, including such as the person you interviewed just a few minutes ago, Joe Heitman.
Have you been able to
sitting right
collaborate?
over there. But nowadays I do an awful lot of work with people who, not that long ago, might not have been all that interested in working with systematists, informaticians and hardcore molecular
What about
geneticists.
the decay?
What about decay?
I think decay is really interesting. So tell
Yeah.
us about what you
Well,
found.
okay, so I mentioned way back in our conversation that Redhead and Gins had split up Lentinus based on decay chemistry. So the brown rot genera, or the brown rot species, were put in a couple of segregate genera, and the white rot things were left aside. And I had studied that from a molecular phylogenetics perspective, mapping on shifts in decay chemistry onto the tree, usually, the methods that you use, trees based on ribosomal sequences, and then you just mapped the characters on. So naturally, when genomics became available, one of the things that I wanted to study was the evolution of white rot and brown rot, but not just making tick marks on cladograms, not just mapping it on, but trying to understand the actual genetic bases of those changes. And that turned out to be an incredibly gratifying area to work in, because we could really see dramatic shifts in gene content associated with shifts from white rot to brown rot. And that's an area I continue to work in now, although now it's not just looking at patterns of transitions between white rot and brown rot, but looking at more sort of downstream questions like the evolution of substrate specificity, and currently collaboration with Jonathan Schilling here at University of Minnesota, looking at evolution of brown rot in terms of shifts in regulation, spatial gene elements of gene regulation in decay fungi. So so that continues. So decay has been great. Morphology continues to be probably one of my favorite subjects, and major focus right now is sort of evo-devo, so evolutionary developmental genetics, again using genomic and transcriptomic methods. And I figure that's going to be one of the major areas I work on for a while.
You gave a wonderful public lecture for the Museum of Natural History, and I remember part of this because you were taking on the observation about the development of the genes for rot in the Carboniferous.
Right.
And you made the point at that lecture that people misinterpreted or misconstrued that data for the headlines.
Yes.
You want to talk about that a little bit?
Sure. So, the most high-profile paper we had on this subject, which was published in Science in 2012, noted that the evolution of white rot, which involves the not just the ability to decay lignin, but that's a key part of it, but also expansion of enzymes that attack crystalline cellulose and other cell wall components. So the elaboration of that white rot mechanism roughly coincided with the decline in coal formation at the end of the Carboniferous, with very broad intervals on the time estimates. So we tried to be rather nuanced in our presentation of those results, but we pointed out that the evolution of white rot may have coincided with this decline in coal deposition at the end of the Carboniferous. And that got out into the blogosphere, and so we started seeing some really silly headlines like coal-killing fungus could boost biofuels, and mushroom culprit for coal shortage. I mean, these were actual headlines. So they completely missed it. And then there was one article in particular that questioned our findings that I think also didn't pick up on some of the nuance of what we were putting out. Anyway, all of this helped convince me that it's really important for us scientists to be able to speak not only to each other but to the general public, and to really engage with the media, because if we don't put energy into telling our stories, the members of the media, even people who are trying very hard, will get things wrong. So that's, I think, part of the reason why I'm trying to write this book now, and why I put a fair amount of energy into speaking to general audiences, such as that group at the Harvard Museum of Natural History.
It was so successful. You know, they came to me after, and they said, "Oh, we filled the hall, and it was being live broadcast, and it was the biggest number that they'd ever had."
And it's because the word mushrooms was in the title. It had nothing to do with me. It's mushrooms.
Well,
and don't give yourself credit.
No, that's just it's a statement of fact. I mean, mushrooms are having this big cultural moment right now,
They are.
and I guarantee that. I mean, I think we broke some kind of attendance record or something. And I guarantee that the overwhelming majority of people in the room had never heard of me, but they were interested in mushrooms. And mushrooms are having this cultural moment, and it may not go on forever, right? So our community has this opportunity right now to, ride this wave and try to support the amateur community and educate them. Because, who knows, 10 years from now, the mushroom moment may be over.
Could be diatoms.
I doubt it, but maybe seaweeds. I doubt that too.
Any other part of your work? We've missed lots of it. The mycorrhizal.
Yeah, so
transition.
morphology, ecology, but the other thing is just classification, taxonomy. It's all about communicating our understanding of diversity in the tree of life. So that's something that matters to me a great deal. I really want, and this really is mainly for other scientists, but it's for all of humanity, because all of humanity is a consumer of taxonomy. I've been very interested in trying to promote understanding of diversity through taxonomy, and also I've been interested in just thinking about how we do taxonomy. I really feel like we're creating a language. We're building the language of biodiversity. Language is something that only works well when everybody understands what it means the same way. So I feel like taxonomy is something that, at least, big picture taxonomy really needs to be done at a community level. It's a communal enterprise, because we're not just creating something for ourselves, we're creating language that everybody has to use. And in fact, so the interview just prior to this one, Joe Heitman, which everybody should go and watch that one, was speaking about the importance of taxonomy in the community that studies Cryptococcus, a very important human pathogen. And there have been some real controversies there about what names to use. So taxonomy really, really matters. And that's something I've thought about a lot. In 2007, we published a large classification paper on a higher level phylogenetic classification for the fungi, which is still my most highly cited paper and probably the most important paper I will ever write. And that was a 70 authors or something trying to come up with a consensus community classification. I'm very proud of that work, and really gratified that so many taxonomists were willing to participate in that.
Well, and follow and use it and follow it.
And there were about probably people from 40 countries, I think.
Yeah, and at the time we had this crazy situation where we all agreed on what the major groups were, but different groups of people who had different major professors were using different names for the same group. So everybody agreed on what the groups were, but they disagreed on what names to use, which is silly. And so that community-driven taxonomy is something I've been interested in, and also other things like automated taxonomy, phylogenetic taxonomy, sequence-based names. These are all things that matter to me because I think that they can have a big impact on understanding of biodiversity and our ability to communicate with each other. So that's been another area that's been very important to me.
Great.
Well, thanks very much. We
Thank you.
enjoyed
Thank
talking
you,
to
David.
Yep, my
and
pleasure.
We'll let you know when the YouTube's up.
Yeah.